Hi, my name is Camila Rodriguez, and today we're going to talk about one part of the hereditary material, which in this case it's DNA. We're going to examine its structure and functions. So, as you may know, all living beings have genetic instructions for the development, growth, reproduction, and functions. All these instructions are carried out in a molecule that, as I said before, is called DNA. But what is DNA? This word that we have always heard during our biology or genetic classes stands for the acronym for deoxyribonucleic acid. But as we have already used this word for a long time, we're gonna explain this topic using the acronym of DNA.
Well, the DNA is a double helix molecule, and this consists of basically nucleotides. Nucleotides are things that look like this. The nucleotides consist of a phosphate group, a sugar group that is also named deoxyribose, and a nitrogenous base. These nucleotides make the nucleic acids that are basically the DNA and the RNA. There are only 4 nucleotides that we have here. The nucleotides are divided by purines and pyrimidites. The purines basically have 2 carbon rings attached and are the adenine and the guanine, and we also have the pyrimidites that only have 1, and these are the thymine and the cytosine.
Deoxyribonucleic acid, or DNA, is a polynucleotide where the strands are formed by the covalent bond between the phosphate group and the hydroxyl group of a sugar, and the steps are basically formed by the bases that are coupled together. That is, it has a structure in which 2 complementary chains are joined together, as you can see here. There is a grammatical rule with these 4 characters: adenine, cytosine, guanine, and thymine. Bases can be written in any order as long as adenine is always paired with thymine and cytosine is always paired with guanine.
Rosalind Franklin was one of the greatest scientists in history. She started working at the King's College London in the biophysics unit. Her experiment consisted of a technique called X-ray diffraction, which allowed the determination of the structure of crystals. She noticed that if she condensed an enough amount of DNA solution, it would separate into fibers, and with certain conditions, it could look like crystals, enabling its structure. The result of this experiment were 2 forms of this molecule, form dry A and form wet B. The B photo of the X-ray diffraction, photograph 51, became really famous as it was one of the critical evidence that was found to prove the structure of the DNA. Unfortunately, she died of cancer at the age of 37 and was never able to be recognized by her intense work and contribution.
Now I'm gonna talk about the Chargaff rules. Erwin Chargaff was an Austro-Hungarian biochemist that emigrated to the United States during the Second World War. During his career, he discovered 2 major rules which help on the discovery of the double helix structure in DNA. According to his first rule, in DNA, the number of cytosine unit is equal to the guanine unit, and the number of adenine unit is equal to the thymine unit. The second rule states that the amount of cytosine, guanine, adenine, and thymine vary from species to species. Also, this hinted that DNA is not only a protein, but could be genetic material.
Watson and Crick's first attempt to build a DNA model without the help of the photograph 51 had 1 success and 1 mistake. The success was that the molecule was organized in racing strands, like the railing of a staircase, and that these strands were formed by the covalent bonds between hydroxyl and deoxyribose sugar rings. This link is called phosphodiester bond because it is the link between the phosphate and the 2 deoxyribose sugar rings. The mistake was that with data from the X-ray diffraction of the 51 photograph, which suggested the DNA molecule was a double helix, and the theory about the bonds that form the structure, Watson and Crick finally made clear the 3-dimensional shape of the DNA molecule, but there was still a piece to put together. The difference in size between pyramids and purines allows the strand to twist in the form of a helix.
The helical arrangement that was established in 1953 facilitated that. Through X-ray diffraction, measurements between bases and between strands could be calculated accurately. That is, the deoxyribose sugars and left strands always go the opposite with respect to those of the right strand. This work is considered the most important of the 20th century because the three-dimensional structure and the arrangement of atoms in DNA molecule explains not only the stability of life, but also the inheritance of intergenerational traits of all species, as well as its mutability so that life continues to exist in a long process of biological adaptation.